Ever stop to think how, the tiniest living things might hold the biggest clues about life itself?
It's pretty incredible, isn't it?
Yeah. Today, we're diving deep into the world of bacteria and these viruses that infect them, bacteriophages. It sounds small scale, maybe, but these little guys, they've been absolutely fundamental to figuring out genetics, how genes recombine, gene structure, even stuff like modern DNA cloning.
It's absolutely true. They give us this amazing window into molecular genetics. And what's really striking is just how simple and fast they are for study. They reproduce so quickly, minutes. Sometimes you get billions of basically identical cells. Plus, they're easy to grow in the lab.
Makes them ideal research subjects, right?
Exactly. Perfect for teasing apart these really complex biological mechanisms.
So that's our mission for this deep dive. We want to unpack the really clever ways bacteria and their viruses swap genetic info and how scientists use these natural processes, through some ingenious experiments, to actually map bacterial chromosomes.
And the principles they uncovered aren't just niche biology facts. They're fundamental to all life.
Yeah, hopefully we'll have a few aha moments today about how heredity actually works. Okay, so let's start with the basics. What makes bacteria tick, and how do scientists even count them?
Well, first off, bacteria are masters of adaptation. They mutate spontaneously all the time, and growing them is surprisingly straightforward. A really key thing for genetic studies is that they're haploid.
Meaning just one copy of their chromosome.
Exactly, unlike us with two copies of most chromosomes. So if a mutation happens in a bacterium, you see its effect immediately. There's no backup copy to sort of mask it. It simplifies the genetics enormously.
And their nutritional needs, that's also a tool for geneticists, right?
Oh, absolutely. We often grow bacteria on what's called a minimal medium. Think of it as like the bare essentials, a carbon source, some salts. Now, a normal wild-type bacterium, we call it a prototroph, it can make everything else it needs from that simple stuff.
But if it has a mutation?
Right. If a mutation stops it from making something vital, say the amino acid histidine, then it's called an auxotroph. His minus auxotroph, for example. It just can't grow on minimal medium unless you add histidine back in.
Ah, okay. So that lets you select for or identify specific genetic changes. Makes sense. And counting these microscopic things, billions of them, how's that done?
That's where serial dilution comes in. It's a classic technique. You start with your liquid culture, could be incredibly dense, and you dilute it down step by step, usually by factors of 10. So 1/10, then 100th, 1,000th, and so on.
Keep diluting until it's really sparse.
Exactly. Then you take a small known volume of one of these really dilute samples and spread it onto a nutrient agar plate. The idea is that the bacteria are now so spread out that individual cells will grow into visible colonies.
And each colony started from just one bacterium.
That's the principle. So you count the number of colonies on a plate from a known dilution, say 12 colonies, from the 10 to the -5 dilution plate.
Then you can calculate back how many were in the original concentrated sample.
Precisely. If you plated, say, 0.1 LML and got 12 colonies from the 10 tough of dilution, you'd calculate 12 divided by 0.1, which is 120 times a 105. That's 1.2 times 107 bacteria per milliliter in the original culture. Simple, but really powerful.
Okay, so we can grow them, we can count them. Now, the really exciting part. How do they get new genetic traits? This is where genetic recombination comes in. It's a bit different from eukaryotes, you said.
Yeah, in bacteria, recombination usually means one cell's chromosome gets bits replaced by DNA from a different genetically distinct cell. The outcome is similar to crossing over in eukaryotes. You get new gene combinations, new genotypes, genetic variation.
And how does this transfer happen?
There are three main ways: conjugation, transformation, and transduction. And a key concept here is horizontal gene transfer.
Right, vertical transfer is parent to offspring, down the generations. Horizontal is sideways, between unrelated cells.
Exactly.
Yeah.
And this is a huge driver of bacterial evolution. Think about antibiotic resistance. A bacterium can pick up a resistance gene from a completely different species through horizontal transfer. It's a massive shortcut to adaptation.
Wow. Yeah, that explains a lot about why resistance spreads so quickly. It's a major medical concern.
A huge concern. It's also a big factor in how new bacterial species even arise.
Okay, let's tackle the first mechanism, conjugation, often called bacterial mating, right? And the big discovery was back in 46.
Yes, Lederberg and Tatum. Before them, the dogma was pretty much that bacteria were asexual clones. Their experiment was, well, elegant. They took two E. coli strains, both auxotrophs. Strain A needed methionine and biotin. Strain B needed threonine, leucine, and thiamine.
So neither could grow on that minimal medium alone.
Correct. But when they mixed the two strains together, let them sit for a bit, and then plated them on minimal medium.
Voila.
They got colonies,
Yeah.
prototrophs. They did. At a low frequency, maybe 1 in 10 million cells, but consistently, they got cells that could grow without any of those supplements. Met plus, bio plus, 3 plus, and so on.
That must have been stunning. Mixing two strains that couldn't grow yields one that can. Proof of genetic exchange.
Absolutely. It showed genetic exchange and recombination had to be happening. Soon after, they figured out it was a one-way street. There were donor cells, called F plus Hay for fertility, and recipient cells, F.
But how did they know it required actual contact?
Oh, that was Bernard Davis and his famous U-tube experiment. Brilliant setup. He put the F plus strain on one side of a U-shaped tube and the F on the other, separated by a filter.
A filter that let the liquid medium pass through, but not the bacteria themselves.
Exactly. So they shared the same growth medium, nutrients could diffuse across, but the cells couldn't touch.
No prototrophs.
None. Zero. It proved physical contact was absolutely necessary. Later, we found out this contact is mediated by a structure called the F pilus, or sex pilus, like a tiny bridge between the cells.
So this F factor, the fertility factor, what exactly is it?
It's a plasmid, a small, circular, double-stranded DNA molecule, separate from the main bacterial chromosome. It's maybe 2% the size of the chromosome. It contains the genes needed for making that pilus and for initiating the DNA transfer process.
And during conjugation, does the whole F factor move across?
Typically, one strand of the F factor DNA peels off and moves into the F cell. Then, both the donor and recipient cells synthesize the complementary strand. So the donor stays F plus A, and the recipient becomes F plus A.
Okay, that makes sense for transferring the F factor itself, but you said the original experiment showed chromosomal gene transfer was rare, like 1 in 10 million. Why so low if F factor transfer is efficient?
Ah, good question. That's because for chromosomal genes to transfer at a high rate, something else needs to happen first. In a very small fraction of those F plus cells, the F factor plasmid doesn't just stay separate, it actually integrates itself into the main bacterial chromosome.
It inserts itself.
Right into the big circular chromosome. And when that happens, the cell is no longer just F plus, it becomes an HFR cell, high-frequency recombination.
HFR. Okay, now this sounds like where the mapping comes in. These were discovered in the 50s.
Yes, by researchers like Cavalli, Sforza, and Hayes. They found these HFR strains, sometimes after treating F plus cells with mutagens, that could transfer chromosomal genes at a much, much higher rate, maybe 1,000 times higher than a standard F plus strain, like 1 in 10,000 cells instead of 1 in 10 million.
1,000 times more efficient. But what was the key difference in the outcome?
Crucially, when an HFR cell conjugates with an F cell, the recipient usually remains F. It very rarely becomes HFR itself.
Unlike the F plus X F cross, where the F almost always became F plus... Okay, that's a big clue. So how did they use this?
This led to the brilliant interrupted mating technique by Woolman and Jacob. Mid-50s, they mixed an HFR strain with an F strain.
Let them start conjugating.
Right. And then at specific time points, say 5 minutes, 10 minutes, 15 minutes, they'd take a sample and stick it in. Get this, a kitchen blender.
A blender? Seriously?
Seriously. A quick whiz in the blender was enough to break the fragile connection, the pilus, between the mating cells, stopping the DNA transfer dead in its tracks. Then they'd plate the recipient cells and see which HFR genes had made it across in that time.
That is incredibly clever. Using time as a variable, what did they find?
It was amazing. They found that genes always transferred in a specific linear order for a given HFR strain. For example, maybe gene disease resistance transferred after 10 minutes, then to resistance at 15, lax utilization at 20, gal utilization at 25, always in that sequence.
A time map. The time it took for a gene to transfer reflected its position on the chromosome relative to the starting point.
Exactly. Time became a measure of genetic distance. Minutes on the map. This was the basis for the very first genetic map of the E. coli chromosome.
But wait, if it's linear transfer, why did you say earlier the chromosome is circular?
Because when they isolated different HFR strains, they found different starting points and sometimes different directions of transfer. But the relative order of nearby genes was always consistent. It was like breaking a circle at different points and reading it out linearly from there.
Got it. So strain HFR1 might start at gene A and transfer ABC. Strain HFR2 might start at gene M and transfer MLK, but the sequence BCD or LKJ would always hold true if they were linked.
Precisely. This pattern strongly suggested the E. coli chromosome was actually a closed circle. The F factor integrates at a random spot, and that spot becomes the origin O for transfer in that particular HFR strain.
And why do the recipients stay F in HFR crosses?
because the F factor itself is integrated into the chromosome, and it's usually the last part to transfer.
Hmm.
Since conjugation is often interrupted naturally or, by a blender before the whole chromosome makes it across, the tail end containing most of the F factor genes rarely gets transferred. So the recipient gets some chromosomal genes, but stays F.
Brilliant. And this technique allowed mapping the whole thing.
The whole E. coli chromosome, yeah. They determined it was about 100 minutes long, conceptually, and initially mapped around 1,000 genes. Now we know there are over 4,000 protein coding sequences.
Okay, there's one more twist with the F factor. This F prime state and merozygotes. Sounds complicated.
It's a neat variation. Sometimes when that integrated F factor in an HFR cell decides to pop back out of the chromosome,
It gets sloppy.
exactly, it can accidentally pick up a piece of the adjacent bacterial chromosome along with it. So now you have an F factor plasmid that's carrying extra bacterial genes. That's called an F factor, F prime.
Like a hybrid plasmid.
Pretty much. And if this F cell conjugates with a regular F cell, it transfers the F factor, including those extra bacterial genes.
So the recipient now has its own complete chromosome plus another copy of those specific genes on the incoming F plasmid.
Correct. It's partially diploid for those particular genes. We call this cell a merozygote. And having these specific genes in two copies, often one wild type and one mutant, in an otherwise haploid cell, turned out to be incredibly useful for studying gene regulation, how genes are turned on and off.
Makes sense. Okay, so the F factor is one type of plasmid. What other kinds are there?
Plasmids are generally these self-replicating circular DNA molecules in the cytoplasm, separate from the main chromosome. The F factor, conferring fertility, is one type. Then there were the really significant ones, medically speaking. R plasmids.
R for resistance.
You got it. These carry genes that make the bacteria resistant to antibiotics. They often have two components, the RTF, or resistance transfer factor, which has the genes needed for conjugation, just like the F factor.
So they can spread themselves.
And one or more R determinants, which are the actual resistance genes. Resistance to tetracyclines, streptomycin, ampicillin, you name it. Even heavy metals like mercury.
Yeah, and they can carry multiple resistance genes on one plasmid.
That's the scary part. A single R plasmid can confer resistance to several different antibiotics simultaneously. The first big scare was in Japan. 1950s, Shigella, the dysentery bug, became resistant to 5 drugs at once because it acquired an R plasmid. These are a major reason for the rise of multi-drug resistant superbugs.
Wow. Are there other types?
Yeah, another group is plasmids. These carry genes for proteins called colosomes.
Collis.
They're toxic to other bacteria that don't have that specific plasmid. It's like bacterial warfare. The plasmid also carries a gene for immunity, so the cell carrying it doesn't poison itself. These usually aren't transferred by conjugation like F or R plasmid, but they show the variety.
And plasmids are also key tools in genetic engineering, right?
Absolutely critical. They're used as vectors to carry foreign DNA into bacteria for cloning and expression, but that's probably a whole other deep dive.
Definitely. Okay, let's shift gears to another mechanism, transformation. This is where bacteria pick up DNA directly from their environment.
That's right. Free-floating DNA fragments, maybe from dead, allyzed bacteria, can be taken up by a living bacterium. If that DNA gets incorporated stably, it leads to genetic change.
Do all bacteria do this?
No, only certain bacteria, or bacteria under specific conditions, are naturally competent, meaning they have the cellular machinery to bind and take up external DNA. It's an active process, requires energy. Usually the DNA binds to receptors on the cell surface. As it enters, one strand of the double helix gets degraded, so only a single strand actually gets inside.
A single strand? Then what happens?
That single strand then looks for a homologous or matching sequence on the recipient cell's chromosome. If it finds one, it can actually displace the original host strand in that region and pair up with the other host strand. Enzymes then kind of snip out the displaced host segment.
So you end up with a small stretch of the chromosome that's a hybrid. One strand original host, one strand new donor DNA.
Exactly. That region is called a heteroduplex. When the cell replicates its DNA before dividing, that heteroduplex unwinds. One daughter cell gets a chromosome derived from the original host strands, and the other gets a chromosome containing the newly integrated donor DNA segment. That second cell is the transformed one.
Can this be used for mapping, too? Like conjugation?
Yes, particularly for genes that are very close together. The DNA fragments taken up during transformation are relatively small, maybe 10,000 to 20,000 nucleotide pairs. If two genes are located close enough to fit on the same fragment, they can be transferred together. This is called co-transformation.
And if two genes are co-transformed frequently, it means they must be physically close on the chromosome.
Precisely. The closer they are, the higher the chance they'll be on the same DNA fragment and get co-transformed. If they're far apart, the chance of them both being picked up and integrated in two separate events is much, much lower. So co-transformation frequency gives you a measure of linkage for closely staged genes.
Okay, one more major mechanism. Transduction. This involves viruses, right? Bacteriophages.
That's the one. Bacteriophages, or just phages, are viruses that specifically infect bacteria. They essentially hijack the bacterium to make more viruses. And in the process, they can accidentally transfer bacterial genes from one host to another.
Tell us about a typical phage like T4. What's its life cycle?
Phage T4 is pretty complex looking. It's got this icosahedral head containing its DNA encoding over 150 genes, and this intricate tail structure with a sheath, base plate, tail fibers. Looks like a lunar lander almost. Its typical life cycle is called the lytic cycle.
Lytic, meaning it breaks open the cell.
Eventually, yes. First, the phage uses its tail fibers to attach or adsorb to the surface of a specific bacterial host. Then it injects its DNA from the head, through the tail, into the bacterial cytoplasm. Almost immediately, it takes over. Host DNA, RNA, and protein synthesis shut down. The host's own DNA often gets chopped up.
Total takeover.
Total takeover. The phage DNA then gets replicated, and the phage genes direct the synthesis of all the phage parts, heads, tails, fibers. These components then self-assemble incredibly into maybe 200 new phage particles inside the dying cell. The final step is lysis. The phage directs the production of an enzyme, lysozyme, which breaks down the bacterial cell wall from the inside, bursting the cell open and releasing all the new phages to find new victims.
That's brutal. How do scientists even count these things if they're just destroying bacteria?
With another clever technique, the plaque assay. It's similar in principle to counting bacterial colonies. You take your phage sample, dilute it serially, just like with bacteria. Then you mix a small amount of a diluted phage sample with a large number of susceptible host bacteria and some melted soft agar.
Okay.
You pour this mixture onto the surface of a solid agar plate. The bacteria multiply and form a continuous layer like a lawn across the plate, but wherever a single phage particle initially landed, it infects a bacterium, lyses it, releases new phages which infect the neighbors, the lias.
And it creates a clear spot in the bacterial lawn.
Exactly. A clear circular area called a plaque. Each plaque represents the descendants of one original phage particle that started an infection. So you count the plaques, factor in the dilution, and you can calculate the original phage concentration.
Neat. So plaques are like inverse colonies. But you mentioned earlier that not all phages just kill immediately.
Right. Some phages, called temperate phages, have an alternative pathway besides lysis. They can enter a state called lysogeny.
Lysogeny sounds more peaceful.
It is, relatively. Instead of replicating wildly, the temperate phage DNA integrates itself into the host bacterium's chromosome, just like the F factor did in HSR cells. Once integrated, the viral DNA is called a prophage.
And just sits there.
Pretty much. It's replicated passively along with the bacterial chromosome every time the cell divides, passed down to daughter cells. No new phages are made, the cell isn't flash-shaped. The bacterium carrying the prophage is called lysogenic.
But can it switch back?
Yes. The prophage can be induced often by some kind of stress to the host cell, like UV light exposure. Induction causes the prophage to excise itself from the host chromosome, and then it enters the lytic cycle, replication, assembly, lysis. So virulent phages only do lysis. Temperate phages can choose lysis or lysogeny.
And this temperate phage behavior led to the discovery of transduction.
It did. This was the Lederberg-Zinder experiment in 1952. Shortly after their conjugation work, they were working with Salmonella oxos this time. Mixed two strains, LA22 and LA2, got prototrophs. Okay, maybe conjugation.
But then they used the U-tube again.
Yep. Put LA22 on one side, LA2 on the other, separated by the filter, and they still got prototrophs, but only on the LA22 side.
Whoa, so not conjugation, because no contact. And they ruled out transformation too.
They did. They added DNase, an enzyme that degrades naked DNA, to the U-tube medium. If it were transformation, naked DNA crossing the filter, DNase should have stopped it, but it didn't. Prototrophs still appeared on the LA22 side, so some filterable agent resistant to DNase must be carrying genes from LA2 across to LA22.
And that agent was?
A temperate bacteriophage called P22, which happened to be carried as a prophage within the LA22 strain they started with. Occasionally, this P22 prophage would get induced in an LA22 cell, start a lytic cycle, and escape. Some of these phages crossed the filter and infected the LA2 cells on the other side.
Okay, but how did that transfer genes back to LA22?
Here's the key. When the new P22 phages were assembling inside the infected LA2 cells, sometimes by mistake, instead of packaging phage DNA into the new phage heads, the assembly machinery would accidentally package a piece of the host, LA2 chromosome.
Ah, so some phages carried bacterial DNA instead of viral DNA.
Exactly. These faulty phages carrying chunks of LA2 DNA, like the wild-type genes LA22 needed, could then cross the filter back to the LA22 side, inject their DNA cargo into an LA22 cell, and if recombination occurred, boom, you get a prototrophic LA22 cell. That's transduction.
The phage acts as an unwitting delivery vehicle for bacterial genes. Amazing. And I bet this can be used for mapping, too.
You bet. Just like co-transformation, if two bacterial genes are close enough together on the chromosome, they might get packaged into the same phage head and transferred together, co-transduction.
Higher frequency of co-transduction means the genes are closer together.
Precisely. It's another powerful tool for fine-scale mapping of bacterial genes that are tightly linked.
So we've covered conjugation, transformation, transduction, these incredibly intricate ways bacteria and phages swap genes. But stepping back, why is understanding all this microscopic detail so critical today?
Oh, it's hugely critical. This fundamental knowledge directly impacts how we understand and fight one of our biggest public health threats, multi-drug resistant bacteria. MDR pathogens.
Just superbugs.
Exactly. We're talking about millions of infections, tens of thousands of deaths just in the U.S. each year. There was that horrifying case in 2016. A woman died from Klebsiella pneumonia, resistant to 26 different antibiotics. Basically everything we had. These mechanisms we've discussed, especially conjugation carrying our plasmids, but also transformation, transduction, are how that resistance spreads so efficiently.
And what's fueling this crisis? Is it just the bacteria evolving?
Well, yes, but we're accelerating it dramatically. Two main drivers. First, massive overuse and misuse of antibiotics, not just in human medicine, but hugely in agriculture. Estimates suggest maybe up to 80% of antibiotics sold are for livestock, often just for growth promotion, not even treating infections. This creates intense selective pressure favoring resistant strains.
We're basically breeding superbugs.
In effect, yes. Second, the pipeline for new antibiotics is running dry. Pharmaceutical companies have largely backed away from antibiotic development. It's just not as profitable, sadly, as drugs for chronic conditions. Development is expensive, treatment courses are short, and resistance emerges quickly, limiting the drug's lifespan.
That's a really worrying combination. Is there any hope, anything emerging from this knowledge? You mentioned phages.
There is. It's fascinating, really. Phage therapy using bacteriophages to kill bacteria was actually explored quite a bit in the early 20th century, before antibiotics became widespread. Then it was mostly abandoned in the West.
But it's making a comeback.
It is. With the rise of antibiotic resistance, researchers are taking a serious new look at phages. Using modern molecular biology tools, we can find, characterize, and sometimes even engineer phages to specifically target pathogenic bacteria, including MDR strains. It's like recruiting the bacteria's natural predator.
Are phage therapies actually being used?
It's still early days in terms of widespread approval, especially in the US and Europe. No phages are approved for systemic human use yet, but some are approved for limited applications, like spraying on foods to control listeria, and quite a few are in clinical trials for various infections. There's a real sense that we might be entering the age of the phage. It's a promising avenue, but it also highlights these really complex ethical and societal issues, doesn't it? How do we balance treating sick patients now versus preserving antibiotic effectiveness for the future? How do we incentivize development of vital drugs like antibiotics when the market forces aren't aligned with public health needs?
Definitely not easy questions. What an incredible tour, though. From seeing how a single bacterium forms a colony to understanding how these viruses can literally pick up and move genes around, reshaping entire bacterial populations and impacting global health, it's just mind-boggling.
It really underscores how studying these seemingly simple organisms has given us such profound insights into the fundamental workings of life, with huge implications for medicine, for agriculture, for everything.
Yeah, and it makes you think, this constant battle, this arms race between bacteria and phages, swapping genes, evolving, it's been going on for billions of years. And now we're learning how to maybe tap into that ancient dynamic to tackle modern problems like superbugs. What other secrets might these ancient conflicts hold for future challenges?
That's a great thought to ponder. We really hope this deep dive sparks some curiosity for you.
Absolutely. Thanks so much for joining us on the deep dive. Until next time, keep exploring.